My name is Aaron Hagar, and I'm the Extension Weed Scientist here at the University of Illinois in Urbana-Champaign. And I'd like to take the opportunity now to thank everyone for taking time to view this presentation on resistance to Group 15 herbicides. And the species we're most concerned about for this presentation is Waterhemp, certainly one of our driver species across a very large number of acres, not only here in Illinois, but across many parts of the Midwest agricultural systems. And the slide that I'm looking at right now, I titled this slide about 13 years ago. I call it the Waterhemp Conundrum. And I've used this slide now pretty much unchanged until about 2 years ago. And so the first iteration of this slide, I simply asked the question, how do you manage a weed population for which there might not be any viable post-emergence herbicide options for its control? And again, we made this presentation with this slide for about 11 consecutive years, but then I had to modify this slide a little bit. And I added the last couple of lines and reduced residual control from many soil-applied herbicides, simply because we're learning new things about different types of resistance mechanisms in water hemp. And certainly now, for many, many years, we have considered resistance and herbicide resistance to many of the foliar applied herbicides. But certainly now we're in an era where we also need to consider resistance to soil applied herbicides as well. And water hemp is a species that more or less has its own rule book. And we really have to understand a couple of important facets of its biology to help us understand how resistance evolves in this species. And the first one, of course, is that water hemp is a dioecious species. And what that really means is that any water hemp plant that you find in the field is either a male plant or a female plant. Male plants produce the pollen, female plants produce the seed. But in order for there to be seed production, there must be pollination. And so with this type of biology, we have a tremendous amount of mixing of genetic recombination that takes place. Females can produce large amounts of seed. There's actually published literature that shows that a single individual female in non-competitive environments is capable of producing in excess of 1 million seeds. And so if you think about this from a biological diversity standpoint, there's no law of nature that says all the pollen needed to fertilize all the flowers on a female has to come from one male. It doesn't work that way. She can receive pollen from dozens of different males. So think again in terms of genetic diversity, water hemp is very, very diverse as a species. And one way that this diversity has been illustrated repeatedly is in the evolution of herbicide resistance. Now, here in Illinois, we have documented resistance to herbicides now from 7 different site of action classes. Some of the initial resistance that we discovered many, many years ago included resistance to things like the ALS inhibitors or the triazine herbicides. And since those initial discoveries, we've also confirmed resistance now to herbicides that inhibit the PPO enzyme. We have resistance to glyphosate, the HPPD inhibitors, the oxinic herbicides, and of course, the topic of conversation here today, the Group 15, or what we call the VLCFA inhibitors. And if you're curious about what this class is, this is actually a very old class of herbicides, first discovered clear back in the 1950s. We're talking here about active ingredients that have pre-emergence activity, and they target the VLCFA elongases. And what that stands for is very long chain fatty acids. And once these elongases are inhibited, essentially the plants starve for these very long chain fatty acids. These are essential components for the formation of cuticular waxes and the cellular membranes. And so sensitive plants really either fail to emerge from the soil, or if they do, they remain in more or less an arrested state of growth after they emerge. So to give you an idea of what some of the commercial products are in this Group 15 or VLCFA class of herbicides, things like Harness and Warrant, Outlook, Dual 2 Magnum, and Zidua would be classic examples of these types of Group 15 herbicides. Now, we say they're old to chemistry, first discovered back in the 1950s, but they're still very important components of our chemical weed management systems in corn and soybean here in Illinois. We initially really recommended these types of products primarily for control of annual grass species. Very good activity on many of the foxtails and the panicums, for example. But as we've seen an increase in problems caused by some of these pigweed species, in particular waterhemp and palmer amaranth, these products have also become very important tools to try to manage these two dicot weed species. They're components of residual control in many of the herbicide premixes, especially those that are used for soybean. And these are very important products as we get into the concept and practice of using layered residual herbicide programs in soybean. So in other words, we're adding a group 15 herbicide into some of the other post-emergence herbicides that are used in soybeans with the idea of trying to extend the soil residual activity for a few more weeks after the application is made. And resistance actually is fairly rare to group 15 herbicides. There are a few grass species. I think in total there's about 13 species worldwide that to date have been documented resistant to group 15. But there's only about 3 broadleaf species that have demonstrated resistance to this class of herbicides. And of the 3 broadleaf species, one is waterhemp and the other is palmer amaranth. So a little background on how we've made this discovery of resistance to group 15 herbicides. Quite honestly, we didn't go out and find resistance to group 15 by looking for it. We first had an indication of reduced control of a waterhemp population that we subsequently confirmed resistant to the HPPD inhibitors, things like the Lotus and the Callisto and the Impact products. And what we did in the field work is we actually had experiments where we evaluated the response of this HPPD resistant population to various soil applied products. And we noticed that the level of control with some of these group 15 products on that particular HPPD resistant population was much less than we normally associate. We really didn't follow up too closely on that until we found another population from here in Champaign County that also demonstrated poor control with HPPD inhibitors. And it's more or less on a whim, we designed a very small field trial for this Champaign County location to look at the response to about 2 or 3 of these group 15 herbicides. And lo and behold, we found a great deal of similarity between how the Champaign County population failed to respond to group 15, very similar to how the first HPPD resistant population responded very poorly. And so in essence, to give you an idea what we see on these populations, if you look first at the lower row of photographs, you'll notice here on the far left, we have the untreated plots from Urbana. And this gives you an idea of what the density of the waterhemp population here is. This is a sensitive population. And as you move from left to right, you can see the level of control that we had from Harness, from Dual 2 Magnum, from Outlook, and from Zidua. Now, in some of these plots, you'll notice that there are a few velvet leaf plants in here. But overall, I think this is fairly representative of what we typically see with group 15 products on a sensitive waterhemp population. But if you now look at the photographs in the top row, this is the Champaign County resistant population. And again, over in the far left, you see the non-treated, and then you see the response of this population to those same active ingredients that we looked at from the sensitive Urbana population. And across the board, you'll notice that the level of control that we see is much less on this resistant population. So that was a graphic representation of what we typically see in the field. And this was about 28 days after application. If you'd like to put some numbers to what we see, this is actually data that are pooled over about 3 growing seasons, again, with the sensitive and resistant populations. And you'll see the various types of group 15 products that were evaluated on these populations. If you look at the far right column, these are the data for the sensitive population at Urbana. And essentially all the group 15 products, again, performed about as we expected to. All except the non-resolved metolachlor formulation gave us over 90% control of the sensitive population at 28 days after planting. Whereas on the resistant population, you can see the level of control with most of these group 15 herbicides is much less than what we see on a corresponding sensitive population. But you really can't confirm herbicide resistance based only on field experiments. You never really know for sure what percent of your population is sensitive and what percent of the population might still be sensitive to these inhibitors. And so to confirm resistance, we move into the greenhouse and we do what's called a dose response experiment. And in a dose response experiment, we're essentially comparing the response of our suspect resistant population to the response of a known sensitive population, looking at various group 15 herbicides, each of which are applied at various rates. And what we typically try to do is we have a range of rates that we'll apply. Sometimes this may be 6 rates. Sometimes it could be as many as 8 to 10 different rates. We start at a rate that's really kind of ridiculously small. And our high rate typically is one that would exceed what the label recommended rates are for a particular soil type. And what we're trying to, again, to do is look and see what the response is of our suspect population now when we do know that we have a known sensitive population for comparison. So to give you an idea of the rates that we looked at and evaluated in our greenhouse dose response experiments, you can see, for example, that the rate of dual 2 Magnum, the low rate was only 0.125 fluid ounce. That was our low rate, whereas our high rate was 1 gallon to the acre. And so again, we apply these range of rates and we're looking to see what the response is of our suspect population and our sensitive populations. And this is typically what we have observed. So to set up this slide, what you'll see is that we have the water hemp plants that are grown here in these small containers, these small greenhouse pots. And we have 4 populations represented in this photograph right here. The population that's called CHRM6 from left to right, and MCRNH40, those are the 2 resistant populations. Those that are labeled WUS and ACR, those are 2 known sensitive populations. And so the populations go from left to right in rows across the slide. In the columns, you'll see the various herbicide rates that were applied to these pots. So on the far left, we have the non-treated. And as you'll notice, the rates increase as you go from left to right. And for example, the column of pots on the far right side would have received the highest dose of, this is dual 2 Magnum. And so if you'll notice what we typically see with the response, if you first look at the 2 resistant populations, again, those labeled in red, going from left to right in the 2 top rows, even at the highest rate, if you look very closely, that rate of 8.4 kilogram per hectare, you can still see some small seedlings have emerged in these treated pots. Whereas if you look at the 2 sensitive populations, we're essentially getting complete control of these populations at a rate that's far less than what we typically would apply in the field. We use about a 2.5 pint rate of dual 2 Magnum as what we call a 1x rate, but you can see that there's many plants in the 2 resistant populations that have emerged at this rate. Whereas again, we have complete control of the 2 sensitive populations, even at rates much lower than that 1x rate. And so what we can do once we have those types of observations, we can now generate some curves. And the curves we generate are used to calculate what's called an effective dose. So we'd like to get an idea and try to quantify more or less how resistant are these plants. Are they really, really super resistant, or are they resistant just enough to escape from a field use rate? And so we generate the curves that you see over on the left-hand side in the panels labeled A and B. The metrics that we look at, A panel, for example, is dry biomass. The other panel in B is actually survival. So this would actually be what we looked at in the preceding slide, the number of plants that emerged from these treated pots. And if you look at now at the table, we have our 2 populations again in red, the resistant populations. And at the bottom, we have the 2 sensitive populations. And the column that we typically talk about when we talk about group 15 resistance is the one labeled the LD50. So that rate that effectively controlled at least 50% of the population. And the numbers that you see within these tables, these are simply the rates that it took to control 50% of this population. So for example, for our two sensitive populations, one labeled WUS, that rate was only 101 grams. ACR, it only took 53 grams to control 50% of that population. But comparing those numbers to CHRM6, it took over 1,800 grams to control that population, whereas with MCR, it took over 3,300. And so if we do a simple mathematical operation by dividing these numbers, we get what's called our resistance ratio. And again, that's an idea of how resistant these populations actually are. And so for CHR, for M6, if we take the 1,800 grams, and if we divide that by the 100 grams for WUS, that gives us the resistant ratio that you see in the third column of 18. If we divide 1,800 by 53 grams for ACR, that gives us a resistance ratio of 34. So in other words, CHR is anywhere from 18 to 34-fold more resistant compared with our two sensitive populations. This was the data that we generated with dual 2 Magnum, but we also did the exact same experiments with several of these other group 15 herbicides. And here you're looking at the results that we had with harness. At our 2.5 pint rate, clear over on the far right-hand column of pots, we essentially had fairly good control of both of these populations. But again, if we start moving now at the rates toward the left, we can still see that there's a shift in the rates. So in other words, we can have some emergence of these plants anywhere from this 250 to 800 gram rate of the resistant populations, whereas the sensitives are still controlled at those rates. Doing the exact same exercise now, looking at these curves, we again generate our resistance ratios. And for acetochlor, for example, we can say that we have resistance ratios here anywhere from 4.5 to 18. We did the exact same work now. You're looking at outlook here at 21 days after treatment. Again, notice the two populations toward the top of the slide. We have emergence that has taken place even at the highest rates, whereas the two sensitive populations labeled WUS and ACR, we still have complete control at rates that are much less than our 1X rate. And again, we see the exact same trend here now looking at control of these populations with Zidua. Now, some might be tempted to look at these numbers and say, well, the simplest solution to this is simply to switch from dual 2 Magnum to harness. But there's a problem with that. And we'll discuss this later as we look at the implications of this. But what we really need to focus on is the fact that there is resistance to all of these group 15 herbicides that we have evaluated. So we can generate these curves for every one of these 4 group 15 products. What is different is the ratios. And you'll see that we tend to see the highest resistance ratios for dual 2 Magnum, whereas we see the lowest resistance ratios for things, for example, like harness. But again, resistance is there to all these group 15s. The ratios are really what separates the products out. Now, we were able to confirm that we had resistance based on our greenhouse and field observations, but we also wanted to know, how is this resistance actually manifest? And so from the greenhouse, we then had to move into the analytical laboratory and do different types of experiments to try to determine what is the mechanism of resistance. And so what you're looking at here, this is actually some laboratory work using HPLC experiments. And if you look at the curves over here on the left, you're looking at the metabolism or the breakdown of dual 2 Magnum in 4 different populations. We have the 2 resistant populations, we have the 2 sensitive populations that we looked at from our greenhouse results, but we also now included corn. And corn is represented by the yellow dots over here on this curve. And essentially what that shows us is that corn is able to rapidly metabolize or break down dual 2 Magnum. That's really why we have selectivity of corn to group 15 products, is that corn can rapidly metabolize these herbicides. Might be a little bit easier to look at the data table over here. And what we're looking at now, again, we're looking at now the amount of time it takes for each of these populations to break down 50% of the applied herbicide. And so you'll see the populations again, ACR, WUS, those are the two sensitive. CHR, SIR are the two resistant. And the other species that we included in this work is corn, because we know corn is naturally tolerant to things like dual 2 Magnum. And so the DT50 column, what we're looking at here, these numbers represent hours. This is hours after treatment for these various populations to degrade or metabolize 50% of the applied herbicide. And it might be easier if we start at the bottom of this table. If we look at corn, for corn, it took 1.7 hours for a corn plant to metabolize or break down 50% of the applied herbicide. If you look at the CHR and SIR populations, they are identical to corn. So in other words, these two populations can metabolize dual 2 Magnum as rapidly as corn can. Whereas the two sensitive populations, ACR, WUS, it takes closer to 3 hours. And so that's why we see this differential response, is that we now know that these populations are resistant because they're able to rapidly metabolize the group 15 herbicides. So to summarize what we've been able to document thus far is that we have at least these two populations that are known resistant to group 15 herbicides. And based on our work with S-metolachlor, we know the mechanism that these populations used is a non-target site mechanism. It's actually enhanced metabolism relative to these sensitive populations. And it's a bit worrisome that these two populations actually can metabolize some of these group 15 herbicides as rapidly as corn. And some other work that we've been able to accomplish with these populations is that they're not using just one metabolic pathway. They're actually using two different metabolic pathways in order to rapidly break down these group 15 herbicides. And that's something that we really haven't run into or encountered previously working with these types of resistance mechanisms. So what could be some implications now of resistance to group 15 herbicides? Well, again, we know there's at least two populations here in Illinois that are resistant, but we really have no idea how widespread or how common this actually is. The take-home message really is that the efficacy now of these group 15 herbicides is going to be decreased. This will only increase over time. We'll find more and more populations over time. Once we typically discover a new type of resistance, it only increases from that point forward. We may need to think about implementing earlier post-emergence herbicide applications if the length of our residual control is not going to be as long with resistant populations compared with sensitive populations. But remember, of course, that not all herbicide failures are due to resistance, and that can be a little bit complicated to work this out, especially as we're talking about resistance now to herbicides that are primarily soil applied. So some other practical ways to consider what these findings represent. Here you're looking simply at a graph of herbicide degradation in the soil over time. Across the x-axis, you can see this is weeks after application. Over on the y-axis or the vertical axis, you will see that these numbers represent the percent herbicide remaining in the soil. And so at 0 weeks after application, that would be the day that the herbicide was applied, we have 100% remaining. But notice over time, that concentration never increases. It only decreases. And it decreases in such a way that eventually we will see that we'll have weed emergence. Rarely do we have any soil residual herbicides that will last the entire growing season. So now we'll see over time when we reach a certain concentration, we'll have the emergence of a sensitive biotype. But if you think about now resistance to soil applied herbicide, it begs the question, well, in relation to a sensitive biotype, when would a resistant biotype emerge? And of course, it's going to emerge at a concentration that is higher than the concentration that's required for emergence of the sensitive biotype. So maybe this will help clarify that a little bit. What you're looking at now are two populations of waterhemp that are actually resistant to the herbicide Valor. The pots that you see in front are sensitive. The pots that you see in the back are from the resistant population. The numbers above these pots represent the rate of Valor that was sprayed on each of these pots. Now, it's not uncommon for soils in Illinois to have an application of between 2 and 3 ounces of Valor per acre. If you look at the 2 pots on the far right of this slide, both the resistant and the sensitive are controlled at that rate. But if you ask the question, well, what is the rate of Valor that's needed to actually control a germinating seedling at any one point during the growing season? That rate is 0.03 ounces. So in other words, you can see at that rate, sensitive is still being controlled. But yet that's a rate that's 100 times less than a typical field rate. So why in the world are we applying a herbicide at a rate that's perhaps 100 times higher than what we need to control the emergence of any particular weed during season? And of course, the answer is we apply the rates that are higher simply to try to get that residual control. We don't spray 3 ounces of Valor, for example, anticipating only a day's worth of control. We apply these rates hoping to get 3 to 4 to 5, perhaps even 6 weeks of residual control. And so now when we go back and look at this graph, when you have resistance now to soil applied herbicides, this is typically the response that we're going to see. We have the resistant biotypes that will emerge much sooner in the season compared with a sensitive biotype. And if you remember back, we hit upon what some folks have said, well, maybe all we need to do with group 15 resistance is switch products, maybe go from dual tube Magnum to a harness type of a product. Well, again, remember that even if you do that, the first water hemp plants, if they are resistant, if this population that you're treating is resistant in this field, the first ones that are going to emerge, even from the harness treated areas, will be resistant ones. Now in water hemp, again, we illustrate the fact that it's a very genetically diverse species and it has evolved resistance to herbicides now from 7 different sites of action. And our weed science program here at the University of Illinois for years has really worked to try to understand what we call target site-based resistance. And that's where there's actually been a physical change in the herbicide target site that renders that plant now resistant to herbicide. So for example, if you look at the right side here, this is the evolution or the time scale when various types of target site resistance has been discovered. It dates back clear to the 1990s when we were able to show that much of resistance to ALS herbicides was due to a target site mutation. But really our group now has really not worked in the area of target site resistance for quite a while. We're now more or less working over on the left-hand slide in the non-target site resistance. There's various types of non-target site resistance, but the one that has most concern, I believe, is the example that we have given here with the group 15 resistance, metabolic resistance, or the ability of a resistant plant to rapidly break down the herbicide. So if we have 7 different types of resistance known in Illinois water hemp, we can show 4 of those 7 have been shown to be due to changes in target sites. But 6 out of the 7, we can also now show are due to non-target site mechanisms. And again, more times than not, we're talking about enhanced metabolism of these herbicides. The challenge with metabolic herbicide resistance is that we really have no predictability left. We don't know which herbicides may or may not still be effective on a population that has different types of metabolic resistance to that. And that is the significant challenge of non-target site and metabolic resistance. How do we manage this? I'll be completely honest with you. We really don't know from a chemical standpoint how to best manage these metabolic resistant populations. We still, in all honesty, are not sure why these metabolic pathways have evolved in water hemp the way that they have. And if we don't know if we're looking at something that's the outcome of using, for example, triazine herbicides for 60 plus years, is it because we've used the group 15 herbicides for well over 50 years? We really don't know for sure. So rotation is not going to be, herbicide rotation is not going to be particularly effective. We've already known based on previous work that even in the absence of a herbicide over several generations, these metabolic pathways, they're still present once that herbicide is reintroduced into the system. Many years ago, our group showed that herbicide Mixing was very effective for target site-based resistance, but it's going to be less so for non-target site because, again, what remains effective can vary by the population. What could remain effective in one field that a farmer has may or may not be effective in the second or third field that that farmer has also. Could there be strategies that overcome this enhanced metabolism? Could there be a synergist? From a molecular standpoint, could we silence some of these genes and then reintroduce them back into the population? Again, a lot of questions that we have unanswered here. How about non-herbicide strategies? Do we need to remove existing plants before they produce seed at the end of the growing season? And the answer to that question is certainly. And I've used this particular slide now for many, many years. This is simply an abstract of work that was done here many, many years ago, looking at various tillage systems and how that affected the seed persistence of water hemp in the soil seed bank. Now, in essence, how this experiment was set up in the fall of 1997, we had an area here on the farm that literally was so overrun with water hemp that you could not see the cornfield that it was growing in. And at the end of the 1997 growing season, all of that water hemp seed went right back into the soil seed bank. During the course of this experiment that lasted 4 years, the key take home is not necessarily the types of tillage that were evaluated, but the key takeaway is that after 1997, no water hemp seed was allowed to be produced. So in other words, there was no seed production at the end of the growing season. And the authors of this experiment went back in and resampled that seed bank each of the 4 consecutive years. And in only 4 years, the amount of that original seed bank that had persisted was 0.004%. That is the weakness of water hemp. That is the most vulnerable stage in the life cycle of water hemp. The most vulnerable stage is not a 2-inch plant that we're trying to control early with a foliar application. The most vulnerable stage is the seed in the seed bank. And it's vulnerable because water hemp seed does not persist indefinitely in the seed bank. On average, you're probably looking at somewhere around 7 to 10 years, and then most of the viability of that seed is gone. So how can you use that information trying to better manage these resistant populations? If a grower can do whatever it takes to ensure that at the end of the season, that there's no seed produced by the female water hemp plants, if you can maintain that practice for 3 consecutive years, 4 consecutive years, you will quickly notice that the population begins to decline very, very rapidly. That is the weakness. Simply waiting for one of the herbicide manufacturers to reintroduce a new active ingredient that's effective on water hemp, that's not going to be the long-term strategy. In other words, we're going to continue to use herbicides to try to manage populations like water hemp, like a 5-way or a 6-way resistant population. But again, the evolution of these resistance mechanisms is going to continue as long as there's seed that's produced by the end of the growing season. So in addition to herbicides, what other non-chemical tactics can an individual farmer implement to try to ensure that by the end of the year, there's no seed? That's the win. That's the victory against herbicide resistance in water hemp.